Hey everyone, thanks for joining us today. I invited Dr. Jessica Rutkoski to the Chestnut Chat because I was reading some papers and I was looking for examples of other people doing breeding for quantitative complex traits and that have used successfully a method called recurrent selection to incrementally improve these traits. So with chestnut, we're trying to improve blight resistance, which is probably controlled by hundreds of different parts of the genome. Phytophthora, also somewhat complex, maybe slightly less complex than blight, and height growth, which are all complicated traits. And the most effective methods so far for doing this kind of improvement of populations for these type of traits is breeding the best individuals for each of those traits and selecting progeny that are even better than the parents. So that's what recurrent selection is. And Jessica, also she understands the foundation of doing this. So like doing evaluating your lines, whatever you're interested in multiple environments, making sure you have good phenotyping methods that are able to really evaluate those traits that you're interested in. And then layered on top of that is doing genomic testing and being able to speed up the selection process with predicting the trait values with DNA sequencing, and also using high throughput phenotyping, like ways of like using drones or other types of methods to look at proxies for the traits that we're interested in that help us do the prediction better. So I'm very curious. I wanted to have Jessica kind of provide some examples of like how this has worked in other systems, like in crops where people have been working on this longer than in trees. And so if we can learn anything from this, and sort of just provide an example of like this works and just give us confidence that we're on the right track with what we're trying to incorporate with Chestnut. And Michael, I don't know if you wanted to say anything about today's Chestnut Chat. Michael's our CEO.
Learn from everything. Thank you so much for the introduction, Jared. Sorry I was a minute late. I was actually just awarding an award to our West Virginia winner of one of our partnership awards. So anyway, I'm looking forward to the talk, and thank you so much.
So without further ado, Jessica, let's hear what you have to say.
Great. Yeah, thanks for the opportunity today to chat with everybody. So my talk is entitled Balancing High-Flying Ideas and Grounded Methods for Faster Genetic Gain. And this is something I've been working on for probably the past 10, 15 years. So, hey, can you see my screen?
Yes, that looks perfect.
All right, great. So to start off, I want to kind of sit back and marvel at what we've been able to achieve in terms of crop improvement over the past 40 years. These are the crop yield trends since 1981 to 2022. This is already in the era of climate change. And you can see that even in that context, we're still seeing really good gains, really good growth in crop yields. Corn yields have improved by 100% over the past 40 years. Wheat, it's been up by 50%, and rice and soybean a little less, but still up by 25%. So think about that. Pretty fascinating that we're still able to achieve this growth. Obviously, it's due not just to genetics, but also management. But keep in mind, the environment has been getting probably less conducive to crop production. So still amazing progress that we've been able to make. And then another example, I think, of amazing progress that we don't often get to sit back and appreciate is just domestication, right? So we, in the past 10,000 years, created entirely new plant species and changed, or just modified these species, making them practically unrecognizable to what they were in the past. So here's some pictures, just examples showing that the modern crops alongside their wild ancestors. So here, the teosinte with maize, you got rice and its ancestor, some ancestral tomato versus modern tomato, and sunflower here. So we don't often sit back and appreciate that, but it is quite a big achievement and had a huge impact on society. Now, the question is, how is all this genetic improvement possible? It seems almost like miraculous or just amazing, right? But so if you ask any person off the street, if you go off or, say your family members during Thanksgiving, I know if I were to ask my dad, for instance, he'd probably say, oh, that has to be aliens, right? That there's no way. How did this happen in 10,000 years? It's just no way. This is some kind of miracle or some kind of um, something came from outer space, right? But we know that's not true, right? So that's not how all this genetic improvement came about. But if you ask another person off the street, they might say, "Oh, it's got to be transgenics." We frankenfood, we genetically modified our crops, and that's why we got these massive yield improvements, and that's why we were able to change teosinte to corn. But, we know that's also not true. That's not how all this genetic improvement came about. Of course, transgenic technology has contributed, especially in corn and soybean, maybe. Probably not to yield improvement, though, to be honest. But that doesn't explain all the other changes. That doesn't explain the improvement that we've gotten in wheat, because there's no transgenic wheat or there's no transgenic rice. So we know this can't explain this improvements in yield that we've been seeing throughout the years. So the correct answer, how is all this genetic improvement possible? It really has to do with recurrent cycles of selection. So that's really the driver of all this improvement, dating back to domestication and also in our modern era. And so what that entails really is a pretty not sophisticated process of selecting parents, individuals in a population, intermating them together, developing a new population, or what we call breeding materials, evaluating those materials, and then selecting the best ones again, and repeating the process. So really a rather simple, much simpler than, say, genetic or transgenic modification, but a really important process, because that's really the foundation for all the improvement that we know today. And, a little bit more how this population improvement works. It works through improving the population, just like it says, population improvement, right? Improving the population as a whole over multiple cycles. So I have this illustration here with these histograms. So looking right now at cycle 1 here. So the blue, dark blue, will be our starting population where no selection has been performed yet. That's what we call the base population. So that base population is going to be colored in dark blue throughout this graph. And then what we do is say this is the distribution of our trait values. You look at the upper tail here would be like the best individuals, right? So we would select those and then intermate them and form the next cycle of individuals. And that would be the light blue selected population. So from one cycle of selection, so selecting these upper tail individuals, the best individuals, intermating them to get their progeny, which is this light blue, you can see that there's some improvement in the population mean. So we've allowed the best individuals to mate, form this progeny population, which is just slightly improved. And from one cycle of doing that, you can see, well, these distributions, they don't change much. There's a lot of overlap. The dotted line represents the mean. So the means are, still pretty close. It's practically not a noticeable difference. Now, if you do this again, so now take this light blue population, select the best individuals from that upper tail, intermate them, form the next cycle, which is going to be down here in the second row, the one I'm pointing to. Now we have this cycle 2 population. Now, if you compare that with the base population, now you can see a bigger improvement. So this improvement just accumulates. You keep working with your most recent population, selecting the best, making progeny, moving to cycle 3, then to cycle 4, and so on. Now you can really start to see a big difference compared to where you started to where you currently are in your population. So from cycle to cycle, you don't see a whole big differences. And so that can be, in a breeding program, when you're a plant breeder, you practically don't notice genetic gain happening, from year to year. But then when you step back and look over time, you can really start to see all this improvement adding up over time. And that incremental progress gives you some big changes over time. So now I'm going to talk a little bit of some genetic trends or improvements in populations through breeding, through recurrent selection, from examples that I've been involved in from my research. So when I was a PhD student, I worked on improving stem rust resistance in wheat, specifically focusing on quantitative resistance. So stem rust is this disease that is in this picture, upper left picture here. And it was a big problem. It still is a problem, but especially, 10, 15 years ago, there was a new race that emerged. And so there's a big need for more quantitative resistance that was going to be durable to changes in the pathogen. And so I worked on that. I'll talk about that in the upcoming slides. I'll also give you some examples or an example from my work on Fusarium head blight resistance. So that's another quantitative disease resistance, and that's in eastern soft red winter wheat. So in the eastern US, this disease pictured here, the symptoms on the grains in this lower picture, it's a big problem in our region in the eastern US. And that's been a really tricky disease to manage. There is no one, chemical treatment or one gene, or it's a very complex resistance. And so, we've attacked that through breeding over time, and I'll show you some examples of that. And then I'm going to show you examples from my work here at the University of Illinois and the wheat breeding program, and some genetic changes that we have affected in the wheat breeding program. So first, as I mentioned, when I was a student, I worked on looking at recurrent selection for stem rust resistance in spring wheat. And we actually looked at phenotypic selection as well as genomic selection, because that, genomic selection was really coming online at that time. And there was actually, a great deal of skepticism about whether genomic selection would be effective. Would it be effective for this trait? I mean, in theory, it should be, but there had been very little work done to actually demonstrate that. And so I conducted 2 cycles of genomic selection and 1 cycle of phenotypic selection in a wheat population, and evaluated it and it was specifically for the quantitative resistance. So we didn't have any effective major genes for that disease in our population. So this is all based on genes with small effects, and we were able to get a reduction in disease severity, which is this in the panel A. You can see how the populations improved. I had 4 different populations. 2 were selected based on genomic selection, 2 based on phenotypic selection. And you can see in both cases, you get this reduction in severity over 2 years, regardless of how you selected, whether it be genomic or phenotypic selection. One thing that was interesting, which you always have to keep in mind when you're doing recurrent selection, is you have correlated traits to your trait of interest. So in this case, there's a trait that is called pseudo-blackchaff that's correlated with stem rust resistance. So that means when the more resistant varieties tend to have this darkening of, it's like melanin that's produced on the heads and on the stems. And so interestingly, as we improve disease resistance, we actually got an increase in the intensity of pseudo-blackchaff. So that was kind of unexpected, because this trait was really considered to be a monogenic, or just one gene affecting it. So we weren't really expecting to see a kind of like correlated response in that way. So that was pretty interesting, although not a desirable trait really, but just illustrating, how selection changes our populations for a trait of interest as well as correlated traits. So now the other example, I want to talk about genetic gains in scab resistance. So over the past 20 years, there has been this huge investment from the USDA through the U.S. Wheat and Barley Scab Initiative to try to coordinate our breeding efforts and achieve better levels of resistance, especially, well, all across the U.S., but it's a real problem here in the eastern U.S., this is where I'm pictured here. And because, we have wet weather very conducive to this disease. And so through a lot of coordinated breeding efforts, we have worked on this disease. And by coordination, what I mean by that is we've got individual states having their own breeding programs, and every breeding program is selecting for all their traits of interest in addition to scab and making sure they put emphasis on scab resistance. In addition to that, we have a nursery that every year people send material, they send their best material, and then we can exchange that. Well, we get to evaluate it in everyone else's environments, as well as exchange the germplasm, and then everybody can use those materials as parents. So it's a good way to get, help each program make progress on their own, but also to get the best material out to everybody to try to make progress in a more coordinated fashion. And so throughout the years, by looking at that nursery and how the entries in that nursery have changed over the years compared to checks, I was able to show that we've actually reduced the disease significantly over the years. Now, the main trait of interest to us for scab resistance is what we call deoxynivalenol, or DON. So DON is the toxin that the fungus produces. And so that's ultimately the trait of economic importance for farmers. And so that trait is the best indicator of, how much impact we're having to combat this disease. And we've seen that this deoxynivalenol, due to genetic improvement, is decreasing by 0.1 parts per million per year. And so going from, 8 down to, 3, for instance. So that's a great improvement. It's pretty slow, but the just small differences in deoxynivalenol can have a really big impact on profitability. So this is still a great result, and we're still getting improvement through the years. So with time, I calculate that maybe by 40 years, we will have such good resistance that maybe this pathogen won't be a problem in our region anymore. So last example, from my work here in the Illinois wheat breeding program, when I first started, I wanted to look at, where are we at in the program as far as our genetic trends, genetic gain, and this looked at the years from 2001 to 2021, and looking at all of our trial data, using checks to control for any non-genetic trends and environmental variability. And so we looked at all the trial data from the past 20 years or so, and saw nice trends in yield moving up, growing up, as well as, heading time going very slightly down, but hardly any change, and then test weight also improving. So these are good things. We want more yield. We want test weight as an indicator of quality, so we want higher quality. We actually do want slightly earlier varieties, so that's also a favorable result. So we saw these really great trends, and that is really all due to the previous breeder that came before me. So when I came here, I inherited germplasm and a breeding program that was already functioning and already had really great foundations. So it was conducting multi-environment testing. It was doing crosses every year with the best parents, and keeping the population going. So doing these recurrent cycles of selection, and the outcome of that was favorable from what we saw. And that's nice, because that also highlights that these trends can be made, these improvements can be made without the fancy tools. So during this time frame, there had been no genomic selection or high throughput phenotyping, or probably no not a whole lot of markers assisted selection really either. So all this was done through, conventional means, just by having a solid foundation and solid breeding methods based on population improvement. So those are just really great examples of what we can achieve. Now, I want to just highlight a downside of, or not a downside, but a reality that we always have to contend with, is that, environmental trends are often negative. What that means is the environment is constantly putting downward pressure on yield. Not always, not in every environment, but in a lot of environments. So we see, and it may not just be for yield, it can be for other traits. So for example, in Illinois, we saw the environment was moving, test weight was getting worse in our Czech varieties. So that means probably that has to do with diseases evolving, because test weight is often affected by disease. So the environment, can be things like climate change, can be things like pathogens. They're constantly these changes that are putting this downward pressure on productivity or quality or other traits that we're interested in. So that just makes our job a little bit harder as breeders. But we still have to keep going in spite of that. Now, this is an example here on this slide, just to illustrate some environmental trends that I've encountered and seen. I've seen these in many different data sets, but in one case, when I was at Erie, there was a long-term continuous rice cropping experiment where they just grew rice for 60 years in a row with 3 crops of rice per season in these different conditions, no fertilizer, with fertilizer. And here were the environmental trends that we saw in each of the different seasons. And you could see that in most cases, we saw this decline in the yields. And there was only in one situation where like it was stable, but there was this constant decline. And most likely that decline had to do with the cropping system, the continuous rice, so continuous flooding conditions that have been imposed. So it's not only just climate change, it can also be our practices, our, cultural practices, and, maybe not having enough crop rotation and things like that. So it can be soil changes as well. So that, as I said, those things just make our life more difficult as plant breeders. So I think this is a great analogy here where, as a plant breeder, we're trying to make progress. We're trying to, build this sandcastle is like our genetic gain, we're improving our population, right? That's my analogy. We're trying to improve our population, achieve some genetic gain. Meanwhile, the environment just keeps kind of trying to erase the progress that we're making. Not that the environment knows what it's doing or is intentionally doing that, but that's just what happens. And so we just have to basically work harder to overcome that problem. But if we make enough progress, we can still achieve a meaningful result in spite of the challenges imposed by the environment. So just to summarize this first part of my talk, which is a lot of background, is, population improvement. It's a backbone of breeding programs. It's needed to generate cultivars every year. We need to improve the population so we can continue to get cultivars out year after year. It's slow, definitely. But it's a steady and reliable progress. So if you think about, if you remember from when you're a kid reading that book about the tortoise and the hare, and even though the tortoise is really slow, because the tortoise is constantly moving towards the finish line, it still wins at the end of the day because the hare is sometimes just not reliable. And so I like to think of that analogy when you think about breeding versus biotechnology or blue sky science. Blue sky science could be really having a huge potential impact, but it also has a high risk of not working. So breeding is great because it's reliable. You know that after 20 years, you're going to get a certain amount of improvement. And so you know what you're getting yourself into. Now, also, we need population improvement just to combat environmental trends. So if plant breeders just stopped doing their jobs today and said, okay, we're going to stop all breeding, we would actually see yields decline because of these changes. So that's just another reason why we need to keep doing breeding. And some people even call this maintenance breeding. So when we talk about disease resistance, sometimes you just need to maintain resistance because pathogens are evolving. Okay, so here's a good stopping point for questions about all those kind of fundamental things before we move on. So the second part, I'll talk a little bit more about examples from my current breeding program, how we've been improving it. But before moving on, I just want to stop there and get some questions on anything I just talked about.
Okay, we have a couple questions in the chat
Mhm.
so far. So Tom Watson asked, what is phenotypic selection and how is it done?
Yeah, so phenotypic selection is just selecting based on phenotype. So phenotype is something you observe in a population. So say you've got, just imagine you have your backyard, right? And you've got a bunch of plants in the backyard. Maybe they're sunflowers or something. And they're all different. Each one of them is different. So if you just go and say measure each plant for height, for example, and then you select, say, the shorter ones or the taller ones, whatever you want. Let's say you want shorter plants. So you're going to select, the best, shortest 10 plants and then cross them again. Well, that's phenotypic selection. So it's really as simple as that.
So with crops, like, are you actually doing like these large, like for yield, for example,
Mhm.
doing like whole plots of the same line where you're like, all
Mhm.
right, I'm going to harvest
Mhm.
this whole thing,
Yeah.
measure the weight of the grain or whatever, all the things that you had mentioned, put it in 10 different locations and say like this line across those 10 locations is the best based on the whole, like you do a lot of replication. So
Yes.
explain a little bit about that.
Yes, yeah. And we'll get into that. I'll give you some details from the breeding program, but yeah, in there's a lot of different ways to do selection. So that's what I mentioned, the most basic one, phenotypic mass selection, just selecting on single plants. In breeding programs now, there's most of the time we're not doing that. We're replicating, so we're generating lines that we can then replicate because in fields, within a field and in different environments, and that gives us better data to do selection. So, but for some traits that are under very high genetic control, you can actually make progress without doing that, without replicating. You can make progress. Some disease resistance is often a good example of that. If you've got a good, screening nursery of some place where you have uniform disease pressure, you can make progress on just phenotypic mass selection, but in a lot of cases, say you'd want to select for yield, there's a lot of interaction with the environment, there's not a very strong genetic control, then you're going to need probably more replication, generate some lines or clones if you're working with a clonal crop, and then be able to replicate those individuals to get a better accuracy of selection.
Heath Dunlap asks, how much of the seed crop improvements are done by universities versus commercial seed companies? And how much cooperation is there between the two? How much do you cooperate with seed companies?
Yeah. Yeah. So it depends a lot crop by crop, region by region. So the statistic that I most recently heard for wheat is it's like 60% public, 40% private. Now, I think there's probably has changed. It's probably more like 50-50 now. The private is increasing. Um, but and it varies by geography a lot. So, but that's probably what it is. And then in the eastern US where we are, there's definitely more investment by private companies because in our region, it really all depends on the economics of the seed. So in our region, farmers purchase seed more often, whereas in the West, they're not purchasing seed as much. They're more increasing their own seed. And so there's less of a private investment there and more influence or more predominance of public institutions. For since we're at a university, we don't I don't have like a commercial arm to release varieties. So we license our lines, our best lines to companies like seed companies who then bring that to market. They increase seeds and they brand it and put it in bags and sell it to farmers and stuff. So we do work with small seed companies and intermediaries that then get the varieties out.
Okay, so Hill Craddock, one of our longtime collaborators from Tennessee, he is asking, is there a different kind of strategy that you pursue for, let's say, major gene type of resistance, like rust, maybe where like a hypersensitive response or something, a major gene kind of just pathogen recognition, cell suicide, versus necrotrophic fungus where it's quantitative? Like, what are the different strategies that you apply to these like major gene versus quantitative type of traits? Are they the same or do you do something different?
So, okay, for say disease resistance is a great example. So for diseases where you've got major genes, there's oftentimes also minor genes. So like I gave you that example with stem rust, there are major genes that exist, right? But the problem with those is they quickly break down. Pathogen can overcome that single gene really easily. And so it's not a durable resistance. Now, if you have a more quantitative resistance, so basically all diseases have there is quantitative resistance. I can't think of an example where that doesn't exist. So there is going to be quantitative resistance pretty for any disease. And that quantitative resistance is going to be more durable most of the time. So from my point of view, that's what I prefer to chase because I don't want to have to be working on those having my genes failing and having to find new ones. I just want to work on and I'm also as a breeder and like quantitative geneticist, that's what I'm comfortable with as well. So I know there are a lot of breeders that do prefer to chase after those major genes because they can give you some rapid short-term progress. And so ideally, I think the ideal strategy is actually to have both, to build up some quantitative resistance in your population at the same time, bring in some major genes to help help that you can have a variety that has very high levels of resistance. So I think when you have those major genes at your disposal, you can do more. There are other techniques you can use to work with those genes, but at the same time, you shouldn't move away from population improvement. Like you should still continue doing that. Maybe the genes are like an added thing on top you can add to that. Now, like other pathogens, like the one I mentioned, like the fusarium, that one is a quantitative. That one there is no like hypersensitive response type genes, so it's probably similar to that situation with the necrotrophic pathogens. You really it's really all you have is that quantitative resistance. So population improvement is a way to go. There's always going to be, for any quantitative trait, or not any, but for many quantitative traits, there are some major genes, some major QTLs, I would say, not major genes, major QTLs that have, maybe they'll explain 20% of the variance, so they'll explain some. And, you can try to select for those with markers. I don't personally do that because I feel like it's just I'd rather just select on the total variance rather than just, a fraction of it. So, but yeah, there are definitely a lot of options when you have like different tools you can use when you have some major QTLs.
We got one more and then we'll move on.
Okay.
And we will there's a few others in the chat, so
Mhm.
we can
Yeah.
get those into the end. But
Yes, yes.
one question I think is intriguing is, do you think that among these traits like growth, yield, traits that are complex, that have been increasing steadily over time, is there a ceiling to that gain? Like, do you think that they can continue indefinitely, or or is there fixation of the favorable alleles, meaning they're homozygous, every plant has these favorable alleles, and there's less
Mhm.
potential for gain.
Yeah.
It's virtually infinite.
Yeah, that's a great question. So I think in theory, it can be infinite because you have mutations occurring, right? So but it really depends on the population dynamics. So if you have a diverse population, say that you're working with as a plant breeder, and you're very good at maintaining, like not, maintaining sufficient diversity from year to year, um, you can, you're going to fix some alleles in your all your favorable alleles. Some of them are going to get fixed, and that's going to reduce your variation. That's true. And that's actually good. I mean, you want to fix the favorable alleles, but then you've got new mutations every year. You've got new mutations. And so if your population is large enough, you will maintain those new alleles because, when they first emerge, they're at a low frequency. But if your population is large enough, they will increase in frequency and then contribute to your variability in the population. So it's really all about maintaining what we call the inbreeding rate. You want your population inbreeding rate to be not too low, or sorry, not too high. So you don't want to be, for example, if you only selected like two parents every year in your population improvement, right? Then your inbreeding is going to be really high and you're going to lose your new mutations. And you're going to exhaust your variation really quickly. But if you select like a reasonable number of parents and you make sure that those parents are reasonably unrelated, you can maintain your inbreeding rate to be slow, and that's going to allow to the new mutations to come to higher frequency in the population and replace some of the alleles that you're losing through fixation. So, and there's methods that we use in plant breeders to make sure that we're not inbreeding our populations too quickly.
Thank you. That was great to have the kind of
Yeah.
question and answer in the
Yeah.
middle. It's a good
Yeah.
strategy. So let's go on to the high flying ideas section of
Yeah.
it.
Yeah. So I just wanted to now give you example on here at the University of Illinois, how I've kind of attacked the problem. When I started here in 2019. And so as I said, I inherited this functioning breeding program. And then I made some improvements and changes, and then I developed some research. And I just want to explain, what was my approach and what are some of the things we've done. So first of all, I think the key thing for any beginning breeder, if you're inheriting a program or starting a new one, is just to understand the region, like understand the environment that you're serving. So wheat in Illinois, of course, we're not limited just to Illinois, where it's the region as a whole, but focusing in on our specific state as an example, we can look at where's the wheat grown, right? It's grown here in the southern part of the state. It's about 800,000 acres at this point in time. And so you can start to ask yourself, well, why? Why is it here? And one of the reasons why is that in that region, wheat is profitable in the double crop system with soybean. So about 80% of the wheat in Illinois is grown in that double crop system. So what that means is wheat is planted in the fall, it goes dormant in the winter, and then starts growing up again in spring. And then it's harvested in end of June, early July. And then soybean is planted the same day that the wheat's harvested. And then the soybean crop is harvested in the fall. So that means you're getting 2 crops in a season, and that's really helpful for getting higher profitability. And there's also a lot of benefits to having 2 crops in a season to cover the ground, and to, help control soil erosion. So you don't see too much wheat in the northern part of the state because you can't successfully double crop that easily. And also, well, another factor is the soil is very fertile and you get really high corn yields, so it's really hard to compete with corn production. So understanding that, I think is important because you start to realize that fitting into the double crop system is really critical. And if we can get the double crop system to work in more parts of the state, then we may be able to even get wheat on more acres. And so that can benefit, and help us to, improve the sustainability of crop production in our region. So here, just explaining, how wheat soybean double cropping is more profitable than other crops in specifically Southern Illinois. So here is gross profit, revenue minus direct costs. And here we've got wheat double crop soybean in blue, and then other single crops shown, corn in red and soybean in green. And if I was to show wheat as a single crop on this, it would actually be 0 or even negative returns. So just to show, wheat in the double crop system is really competitive with the other crops. And that's really critical to make sure that wheat remains a part of the cropping system. Now, I think for me, like when I look at this, I feel like my goal should be to make sure that this profitability remains high or improve it even further and make this more feasible or more profitable even in other parts of the state. Because if we can do that, we can probably, increase the prevalence of wheat in our region. So with that said, then now my objective is to improve profitability, right? So we look at what are the factors that contribute to that. The factors are yield, test weight, deoxynivalenol, and maturity time. So yield, obviously, it's a no-brainer, right? Test weight contributes because when you have too low of test weight, you get a discount, or the price that the farmer can sell the wheat for gets reduced. Also the same for the deoxynivalenol, which is caused by the scab disease. So we need resistance. So we can have low levels of deoxynivalenol because deoxynivalenol does affect the price of the grain. And then maturity time is important because for the double crop system, the earlier that the wheat matures, the earlier the soybean can be planted, and then you're going to have a higher yield on the soybean crop. So really defining that, okay, what are the traits underlying profitability, and then focusing in on those, I think that's really critical when you're starting or redefining your breeding program. One thing to keep in mind is there's probably thousands of regions in the wheat genome involved in all of these traits. Like when you think about all the traits combined and across, all the environments, the entire target population of environments, you can think there's thousands of regions. So we're dealing with a very quantitative trait, really a quantitative objective, which is profitability. All right, so then, okay, we got the objective. So first, before we think about the strategy, the breeding process, knowing that we're working with quantitative traits, we need to figure out how do we improve genetic gain, or how do we develop a strategy that is going to achieve genetic gain. So here is our formula that tells us what's our expected rate of genetic gain per unit of time based on these key parameters here. So number 1, selection intensity. Selection intensity in orange here. Selection accuracy, which is in purple. Added to genetic standard deviation, and the cycle time. So we want to improve things like selection accuracy and intensity and we want to decrease the cycle time. So using that as a guide, or using those concepts to guide us, we can then think about how do we develop the right strategy to achieve genetic gain for our traits of interest. So this is the current strategy for my wheat breeding program at the University of Illinois. So I've kind of divided it up into 3 types of activities. One is recombination and line development. The second is phenotypic evaluation. And then the third is pure seed production. So recombination and line development, we'll walk through that a little bit, but here colored in yellow are all the processes that happen in the greenhouse. Everything else happens in the field. So we do crossing, generate F1 plants. Then each F1 plant, we plant those seeds in what we call F2 bulks, which are segregating families. And then we harvest F3 seeds and plant them as single plants in the field. From those single plants, we can then increase the seed in a seed increase plot. We also, in that seed increase plot, will perform some selection. So either by phenotypic, or now we're starting to do that more with genomic selection, so based on our predicted breeding values. And then those individuals, after we've selected them from the seed increase plots, we will then put them through phenotypic evaluation. Before I get to the phenotypic evaluation part, I want to just point out, at this point in time, this is really our first opportunity to start selecting individuals that we're going to use again as parents. So that's a critical part. So we can, from this point in time, we have our individual lines at this point in time that before we even start testing them, we can actually select parents and then use them for crossing. So that right there becomes our minimum, breeding cycle time from crossing down to the seed increase plot, the F3 derived F4 seed increase plot. We, based using our genotypic data, we can use genomic selection here and then start selecting parents again. So I'll show you a little bit more details about the recombination and line development, and then later I'll give you some more details on the phenotypic evaluation part. So we'll start with this part. So starting with the crossing, as I mentioned, we have all the individuals that we are crossing with. We're selecting them based on genomic estimated breeding values. So that's combining phenotypic data with our genomic data and getting a genomic estimated breeding value, which tells us how that individual should perform as a parent. And we have a cross prioritization process where we actually have this all like in an algorithm where we have, okay, what's the expected merit of every cross, every combination? And then when we go in our greenhouse, we take inventory, okay, what's ready to be crossed that day, put that into our tablet, and it tells us what's the list of crosses to make every day. So that's something that's really helped us improve efficiency and making sure that we're making the best crosses. So once we make our crosses, if we grow up the F1 plants, so the F1 plants, we harvest those seeds, we put them into these, we call these mini bulks. Actually, that term was developed from Michigan State University, developed this protocol. So we take those seeds, put them in high density planting. We have to vernalize them, which is giving them a cold treatment, transplant them, and then here's what they look like at maturity. Then we harvest those and we get our F3 seeds that then we plant in the field. So we want to accelerate this process as much as possible. Now, in the past, my predecessor did all this in the field, and so everything took a year, before. So it was like crossing was 1 year, F1 1 year, F2 another year, F3 another year. So it was a really long process. And so we've managed to get all these steps down into 1 year. So that really helps us accelerate our line development, which helps us reduce our cycle time that contributes to accelerating genetic gain. So with our bulk seed, we get that seed from the greenhouse, we plant them. Just look over to this part on the right. This is the pipeline we are using. We're planting F3 single plants in the field, and then we go and select single plants, and then the selected ones we increase in our small increase plots. And at that point in time, so anything that we increase, we also genotype. And so that allows us to perform genomic selection at that point. Now, just wanted to, on the left side of the slide here is a comparison, like what's the conventional pipeline look like? Because this pipeline, again, it's updated or improved or, made more rapid compared to conventional. So in a conventional pipeline, you'd actually put F3s in bulk and then take single heads, put these heads in a single row, and then from there, put it in a small increase plot. So we've actually cut out a step. We've cut out the head row step, again, saving us more time. So just emphasizing that we want to shave off as much time from our breeding cycle as possible. Okay, so now we talk a little bit about the phenotypic evaluation. So after we increase the seed here, we have our lines, we start to test in multiple environments. So we'll test for the first year, we'll test in 2 to 3 environments. The next year we'll increase that. We'll actually double the number of environments every year. So we'll test, say we test in 3 environments, well then the next year will be 6 environments, and the next year will be 12, and then 24. And then every year that we're testing, we're removing lines that perform poorly. So if we start out with 1,200, we'll advance a third of those. So we start out with 1,200, then we'll get down to like 300, and then from there 100, and so on. So it's roughly a third, or between a third and a fifth, depending on also the composition of the population. And so we're increasing as we go through the stages of evaluation. We decrease the number of lines and we increase the number of environments. All that data is what we use for making decisions. We make decisions on what to advance, but also we make decisions on what to use as parents as well. So all this information helps us improve our genomic selection models. So we use that to train all of our models. And when we're making selection decisions, we're using all of the data that we've accumulated from the past years of testing in that process. Here, I've just indicated, how long it takes to get to a variety. So with our process moving from line development all the way to phenotypic evaluation, it's 7 years, but we can select parents after about 3 to 4 years. So our breeding cycle is actually 3 to 4 years. So that's because the time that it takes from crossing to identifying a parent of those crosses, from those crosses, that's the cycle time. So that's really what drives the genetic gain. And then you want, every breeder wants to develop varieties more quickly. So that's also a great added bonus when you're shaving off breeding cycle time. So I'll talk a little bit more now, okay, the details when it comes to testing. So here, this is how we evaluate yield. It's a really critical trait. It's the main trait driving profitability. And so we have to get that one right. We invest a great deal in evaluating that trait. So here's one of our field sites. Each one of these plots, which is the rectangle here, is a different line. In this field, at least 20% of the lines are replicated. And then here you can see this is our research plot combine. It goes and harvests a plot, and then it can weigh and take other measurements of the grain as well. Now, as you imagine, this is a really expensive process, not just the harvesting, but all throughout the season. It's very labor intensive and just expensive to conduct. So we do different things to save resources and to be more efficient. So one of the things is what we call a sparse yield testing design. So here on this graph, on the y-axis here is all the locations, and on x here is like all the lines. So all the vertical lines is an individual breeding line, and then you can see where it was tested and whether it was replicated or not. So if it's replicated, it's in blue; not replicated, it's in orange. And you can see majority of the lines tend to not be replicated within a site. That helps us be more efficient. We do have some replication, which helps us to estimate our spatial error and control for those factors. And so all the lines more towards the right, these are lines that are in the later stages of testing. They're going to be more replicated, and they're also in more environments. And you can see another thing you'll notice why it's called sparse testing is you don't have all lines in every single environment, but you have enough overlapping that you can estimate the genetic effects across the entire set of trials. Another thing we do, we actually randomize all of our breeding stages together. Now, that may sound obvious sometimes to some people, but if you look on this on the right of the figure, most breeding programs had been or are currently not randomizing all breeding lines together. They'll just randomize depending on the stage. So all lines in the first year of testing will be together, all lines in the second year of testing together, everything in the third together, and so on. So it's not completely randomized, but that's something we've changed because we are focusing on, improving the design and not having like confounding between environment and genetics. So that's just a detail that we've implemented. We found that actually does help us improve selection accuracy. We've done some simulation work just to confirm that. So we can see here the complete randomization, like what we're doing is in this red, versus the old way, what we call restricted randomization is in blue. So complete randomization gives us a little better genomic selection accuracy, especially when you have less field uniformity. Okay, how am I doing on time?
You have till 1 o'clock, so should leave a little bit of time at the end for questions. We're at 12:28 now.
Okay,
Yeah.
okay, great. So now those things I mentioned, they're still pretty basic, pretty fundamental, focusing on, trial designs, reducing the breeding cycle time. I consider these like the lower hanging fruits of what you can do to improve your breeding program. But now here we've got some more techniques we can incorporate, like more predictive breeding approaches, genomic selection. And maybe genomic selection should be considered a low-hanging fruits at this time, but I'd say, it still can be considered a more advanced a technique depending on, where your crop is and your familiarity with it. And then high-throughput phenotyping is another one that it's kind of still an active area of research that can help. But just to explain a little bit more background what these are. So with genomic selection, we have, phenotypic and genotypic information. We use that to train a statistical model, and then that statistical model is used to predict breeding values. High-throughput phenotyping, somewhat similar, except you're not incorporating the marker data, genotypic data, so you have just your phenotypic data from sensors, and you can use a statistical model again, and then use that to help estimate breeding values. So I'll just show you some examples of how we've been exploring and implementing those different methods. So I'll show you for scab resistance, how we've done that. So with scab resistance evaluation, that's really challenging to do. So we have to do this in the field in a nursery that's inoculated and then measure all these different disease traits, incidence and severity, and the percent of kernel damage and the amount of toxin in the grain. So this is really, it just adds a lot of labor, having to measure all these different traits to get a good evaluation of resistance. So we thought, what if we try genomic selection, specifically multi-trait genomic selection for scab resistance, and potentially we can use the data in just a smarter way to be more efficient. So we tested, okay, so what if we have a set of lines we call the training set, and we have all of our phenotypic data. Our main trait of interest is the toxin, DON. And then we have phenotypes for traits other than DON, and then the marker data. So our training set has everything. And then you've got, our new breeding candidates. Let's say we don't have the DON data, but we have phenotypes for other traits, and we have marker data. So we want to predict those. Okay, so now we tested this approach, and we tried a bunch of different combinations of the phenotype, the other traits other than DON, to see, well, maybe we don't have to phenotype all the traits. Maybe there is just some traits that we can drop. Like maybe we don't have to do all, every trait, and see if we can improve efficiency that way. So here, just some, accuracies of selection. Here we've tried just using our secondary traits, other disease traits. This one that I'm pointing to here is just our accuracy for predicting DON. And then here's all the multi-trait models. So we get a better accuracy with multi-trait models as expected. But one thing that really stands out is that we get some of our best accuracies when we just use one other trait together with DON. So we don't really need to include all these other traits. So what we figured out is we really only need to evaluate two traits, which is fusarium damaged kernels, which is the percent of kernel damage, and the toxin. And then we can select using multi-trait genomic selection. Another thing we found out, which I didn't show results for this on the previous slides, but we also found out when we're using multi-trait genomic selection, we can reduce our replication and still get a good accuracy. So this is helping us be more efficient. Okay, and then last, just trying to knowing that it's important to evaluate fusarium damaged kernels, we want to see, okay, so there's some methods we can use to make this phenotyping better. And so we tried using artificial intelligence to evaluate kernel damage due to scab. And so what I'm showing on the slide is a petri dish, is a bunch of grains. And normally what we would do is just look at this dish and give a subjective rating, like what's the percentage of damaged kernels in this dish. So can be really tricky and tedious and really difficult to do. But yeah, we would try to see, can we use some kind of image analysis to make that better? So we tried that using, this algorithm. It's called mask RCNN. And you have to go through and take images, label individual kernels, and then train the model to recognize what is diseased and healthy. And then so we did that, and then we applied it to a bunch of other images to see how well it could predict the percentage of kernel damage. And interestingly, we still found that visual ratings were still superior to what we could achieve with that AI, algorithm, mask RCNN. So here we're trying to predict deoxynivalenol using our fusarium damaged kernel ratings. So the top here is showing that correlation between the visual ratings from human beings and then the fusarium damaged kernel scores from our algorithm. So we didn't get as good a result with the mask RCNN compared to human beings. But nevertheless, we thought, well, maybe this has some promise for breeding still. So we look, what if we use this in genomic selection? can it help us improve accuracy at all? So here we look at what about the situation? What's the accuracy when we do not observe candidates? We just train the model with our training set, marker data for dawn, and then we try to predict new candidates. So this is not having any observed, kernel image or human raters involved. So this is just the baseline, like what's our prediction accuracy for the dawn? We get our accuracy of 0.26, so pretty low, but this is telling us, the baseline, what we want to improve upon. And then what if we have our phenotypes from mask rCNN and the images? Well, we can increase that quite a bit. Now accuracy gets higher, 0.61. And then how does that compare to with what we'd have with visual ratings? With visual ratings included, accuracy is 0.97. So you can see, as you might expect, we get our best accuracy in a genomic selection approach. We get our best accuracy with the visual ratings of kernel damage versus our mask rCNN. Nevertheless, we still got, mask rCNN was still better than nothing. Okay. So one of the things that we found, obviously, we found visually ratings better than mask rCNN ratings, but still, having any ratings is better than nothing. But what we kind of discovered through this is like you'd really have to have a lot more data and more research before we could replace, human raters for this trait. So they really require some more work. I think it would be possible, but we would just need a lot more data to train on and more time invested in that. So for now, we're sticking with human raters. Okay, so then one of the things, again, now we're trying to see if we can do something similar with yield, where we're looking at, can we use predicted phenotypic traits, like predicted yield from the information we can collect from drones? Can we use that to help us train genomic selection models? So we can take, data from our field, taking data with the drone images like this across the field, and then grain yield for a portion of the plots. Can we use that together to give us, some predicted yields that would then help us improve genomic selection accuracy? So that's what we're trying to do. I've got a couple people doing research on that in my group pictured here in this slide. But it's definitely a long-term research topic like this. Still, it's the methodology hopefully is evolving rapidly. So, but we're not quite there yet on incorporating these kind of tools. So just summarize from part one, population improvement is key. It's the baseline. We need that for sustainable progress. And then predictive breeding methods, they can potentially enhance the efficiency of an already well-functioning breeding program. And I think I want to emphasize, we should really focus on doing what we know how to do today. And there are a lot of tools out there that are not quite ready, for prime time. Those are great for research, but for breeding, you got to stick with what works. So then I just want to acknowledge, this is my team, that every year we have different undergraduate interns. These are some that we've had over the years. Graduate students and staff are here. We also collaborate with other states, breeders in other states, and we also test with farmers on their fields. So I just want to point out it's a really big team effort. And also we've, want to acknowledge all these people and sources of funding that we rely on. So I hope, I think I left time for questions. I hope you all have some good questions and feel free.
Okay, so amazing. I really loved seeing your example, and I definitely want to talk to you more about bringing all these tools into helping chestnut restoration. But a few questions that came in from the chat or the Q&A. These are two related questions. Please define genomic data. And then the second is your version of genomic selection. We're using what's called GBLUP, which is just basically using genomic data to infer relationships between individuals in your breeding population and using the weighted average of the relatives phenotypes to predict the breeding values of the trees or the individuals that were genotyped. So can you explain a little bit about your genotyping methodology and like what's your prediction methodology? Yeah, what works for you?
Yeah. So we're using genotyping by sequencing, not because that's the best platform, but it's the cheapest, and it's good enough. So probably if we had, if SNP chips were cheap enough, that'd probably be what I would do. But I'm going with, I'm trying to be cost effective, and so for GBS, genotyping by sequencing is what we've been doing. So with that, it's just, you sequence, basically the genome gets cut up into fragments that get amplified, and then you sequence that, and then there's a SNP calling pipeline that you call SNPs with that. So that's what we use. I don't do any of that in-house. I send DNA off to a lab in North Carolina, and it's the Small Grains Genotyping Lab, and they do that for us. And
How much
then
does it cost, like per sample about?
so it's about $10 a sample.
Wow, that's
Yeah.
pretty good. Wow.
Yeah. And um, as far as prediction models, we do GBLUP as well. But I use a factor analytic model. So that is to help account for genotype by environment interaction better. So that allows you, and that also is great because it lets you come up with different combinations of environments. So if I say I want to select for just the north, I can generate a BLUP that is just weighting the north environments, or I can generate a BLUP that's only weighting the southern environments, or I can weight environments differently how I want to. So yeah, so it's a variation of BLUP. It's just more a multivariate. It's actually a multivariate BLUP.
So are you using that like environment data to deploy, I guess, and select different lines depending on like whether you're in the south of Illinois versus like a different part of Illinois or different states altogether? Like, are you using that specific kind of prediction and performance and environments to like select different individuals in these different environments?
Yeah, that's more definitely, and that's more towards the end of the pipeline when we're trying to identify varieties. Then that's when it's that regional performance really makes a difference. But when I'm selecting parents, I'm really just considering all of the environments, in the target set. Um, but I still use that same type of model because it just provides also benefits. Like when you have a lot of G by E, it's tends to be better.
Another question from Clark Riley. He's asking about what kinds of acreages are involved in something like wheat or barley breeding. And I guess what I'm expanding on this question, like how big are your cooperative programs and like how many institutions are you working with like to generate some of these large multi-environment trials? Like what's the social dimension of this, like the
Yeah.
social technology of doing this?
Yeah.
Yeah.
Yeah. So all the trials that we conduct in Illinois, we manage, but we rent land from farmers. We test on one experiment station, and all the rest is through farmers, cooperators. So we rent land from them, and then they prepare the field for us, and then we'll plant and harvest. And that's somewhat transactional because we rent the land, so we're paying for it. But with that said, they do, it probably like they have to communicate, and they have to be interested, and like they have to enjoy doing it. Otherwise the money probably isn't really enough because it's kind of a pain in the neck for them to have to consider us in their process. So there's that. And then we also run the statewide wheat variety testing trial. So we're managing all that as well. So some locations are strictly for that and not for breeding. And then for like our out of state cooperators, we have something that we call the Big Six, which is a cooperative trial where we send some preliminary advanced lines. We pool lines together from multiple states and then test them across the region. So that gives us a lot more environments. So I'll test that big trial with, germplasm from different states. And then all the data gets shared back to everybody else. So that's a way to get a lot more environments, especially on your more advanced breeding material. And then I also have a nice um, collaboration with Virginia Tech where we exchange plots. So it's a little bit of a different setup where we're not doing, it's not really cooperative testing. We're just kind of renting each other's land in that sense. So it's like I'll send him 700 packets. He sends me 700 seed packets. I plant them in my big field. He plants mine and his. And then at the end of the season, we swap data. So that's really great. I honestly wish that was done more frequently because it allows everybody to have like that same freedom to operate, but you're just improving efficiency by exchanging labor in a sense.
This is a question for me. So I noticed like you had that whole like you had compressed your breeding cycle down into a year with all of that optimization of growing the plants and doing genomic
Mhm.
testing and all of that. And then you also have you use like multi-trait, like you use proxies for like the trait that's really hard to measure, like that Don.
Mhm.
And so you're improving your accuracy by way of doing this proxy phenotyping in addition to genomic prediction.
Mhm.
So of the two things, like the high throughput phenotyping in addition to your typical phenotyping versus speeding up the cycle, so
Mhm.
you're improving accuracy and you're reducing cycle time, what is like the biggest improvement in terms of gain, slowing this or speeding the cycle or making more accurate selection?
Yeah, so I think the best way to approach it, I think, is look at the biggest area of weakness. So if your selection accuracy is low, then that's your number one priority is to improve that because it doesn't matter how fast you go, if your selection is not effective, you're not going to have progress. So you need to get everything up to an acceptable level. So if you have something that's like unacceptable, you need to work on that. And then if your accuracy is good, but your cycle time is lower, your cycle time is long, then focus on that. So I would say like you want to just find the area of biggest weakness. And then like if your accuracy was like moderately good, then maybe, but your cycle time is long, maybe focus on cycle time, you know? So I don't know if that's a clear answer, but um yeah, it's sort of like just looking for that. Like think about like you've got that bucket of water and like you've got a hole somewhere. You got to fix that first, if you think of it that way.
Yeah, with trees, cycle time obviously
Mhm.
is like the thing that would
Mhm.
probably give us the biggest improvement. Like, and we're
Mhm.
experimenting with um, we inoculate seedlings in their first year rather than waiting in the field for
Mhm.
like 5 years or whatever, and then train genomic prediction models on the seedlings from particular families, like some subset, because the tiny seedlings, like we kill them all, even the resistant ones. But we get good data from a certain window of time. And then
Mhm.
we can use speed breeding techniques like grow them under high light in a greenhouse but still that's still challenging
Mhm.
the tree because there's a large seeded tree, so we're not getting tons of seed progeny. So
Mhm.
just kind of like our context of like
Mhm.
the cycle time is definitely the weakness. Accuracy we can improve as well, but cycle time is really
Right.
challenging. And okay, so one other question, another one from Fred. Can you explain the benefit of waiting for phenotypic selection until F3s as opposed to previous generations? Like, why do you do that? Like, those all the way down to F3. Why not do the selection or phenotyping at those other earlier generations?
Yeah, so since we're still generating a line, and it's like there isn't, and there, well, there's two things. One, we're still generating the lines, so we don't have any ability um, to really replicate or select accurately at that point, and we're doing that in the greenhouse. There really isn't anything that we can select that would be relevant at that point. Now, phenotypically, now it is true that we could potentially select based on genomic selection before generating a line. Like you don't have to generate lines to do GS. You can do that on single plants. So, you know, in theory, we could do that much sooner, but we don't do that. Mainly, it's really a cost thing at this point and a logistics thing because we have to, we want to be able to update the training population. So if we genotype a bunch of candidates that are like F2s, then when we generate lines, we're gonna have to genotype those lines again. So we kind of have double genotyping. So, you know, I don't know. I think there's probably it's probably debatable. Like there are probably people out there that think like, oh, I should probably be doing the genotyping F2s and selecting those. Um, but that's the reason why we haven't done that, just because of, like it would just be a lot more genotyping, and a little bit more like harder to manage.
What Kim Steiner asks, what levels of heritability are we talking about for like traits like yields, Don, all of those, and the ones that you were most interested in your selection indices, like and heritability, just a context.
Yes.
Heritability is percent of the variation in the trait that is genetically controlled. So like
Right.
something like 0.2 is like 20% of the variation is genetic. So what's kind of normal for these kind of
Yeah.
traits?
So yield, you know, on a single plot basis, heritability is like 0.1, you know, on a good day. So it's really low. Um, and what I mean by single plot basis, like if I was to just walk up and measure yield on individual plots and make a selection decision based on that, that's the square of my selection accuracy. So, and then for Dawn, it's probably a little higher. It's not extremely high, around 0.4 across environments. Now that's considering like we're only testing it in our inoculated nursery. So it's not, we're probably not sampling like all the possible G by E that may occur, but it's a little higher than yield. Mhm.
Last question. So I don't know how much you're kind of a student of like breeding techniques beyond like doing what recurrent selection,
Mhm.
but for example, we're using hybridization with Chinese chestnuts to introduce where our assumption and what we see with the data is that there is a long evolutionary history of the Cryphonectria fungus, the blight fungus, living with Chinese chestnuts. They have some genes in their genome that are not present in the American chestnut. American chestnut has variation in for blight resistance, but when we use hybridization, we increase the level of variation and we increase the level of resistance.
Mhm.
So we are using hybridization because I think that's going to give us a bigger gain relative. We are also using recurrent selection in American, but
Mhm.
American chestnut. But so what, in the crop world, like, do you have examples of like hybridization, interspecific crosses, like other things that like have helped with improving gain relative to doing just recurrent selection
Mhm.
within species, like that you could think of that might be some examples that would be helpful.
You know, across species, like the only examples I can think of are like very specific genes, like not necessarily bringing in like, um, you know, a lot of different loci, but um, yeah, that's what I can think of. And honestly, I can't think of specific examples other than like, say, you know, stem rest resistance. I know there's a lot of genes, like major genes from different wild relatives of wheat, and like people have brought those in on like translocations and things like that, and worked with it that way. but it's interesting, like I don't think about tree species that much. So you're taking hybrids, meaning like is the population itself as like a result of those like interspecific crosses, or you have to just generate at the end like a hybrid?
Yeah,
does
so
that
we've done like taking a Chinese chestnut and American chestnut, they're interfertile. You can
Mhm.
cross. We make F1 hybrids, and then we've done back crossing, taking those
Okay.
hybrids and bringing them back crossing them to American,
Yeah.
and with the and we're trying to figure out like what's the level of like Chinese chestnut ancestry versus American chestnut ancestry that gives us acceptable levels of resistance where the trees can live and reproduce. But also they can grow tall because the Chinese tend to be bred for shorter stature for
Yeah.
harvesting from the ground, and the Americans are tall forest trees, and we want the tall forest trees, but we also want the resistance from the Chinese chestnut. So we're trying to seek this balance
Mhm.
between these two things, and they're all the chestnut species are interfertile. You can make hybrids
Okay.
of every kind.
That's interesting. Huh.
Yeah.
Yeah.
So it's a little, it's maybe a unique example of what we're doing is maybe a little bit out of the norm.
Yeah.
But we are definitely needing, when I look at the genomes of the species, like there's definitely stuff in the Chinese genome that's been duplicated or new gene families that are involved in the resistance mechanism, the resistance that are not present in the American chestnut. So that's we're introducing this novel variation and hoping that evolution can help us select. I mean, we get it to a baseline level of resistance, and evolution can take over for us.
And you can recombine the resistance from both
Yes,
species. Yeah.
exactly.
Yeah.
So I think that's it for questions. Really appreciate hearing your examples. I don't know if you have anything you wanted to say at the end for people, but I really appreciate showing what works and giving us an example of how we can orient our breeding program to success.
Yeah. Great. Well, it's a pleasure to talk to this group, and it's interesting to see people so passionate about breeding and what it can do, and just like 89 people showing up to this webinar, like that's amazing. So yeah, just uh awesome to see that.
Well, thank you. Appreciate
Yeah.
it.
Yeah.
Good to see everyone. Thank you for coming. It was like more like 130, 140
Yeah. Yeah.
people came, and they were present for various parts of it.
Nice.
So
Nice.
yeah.
Awesome.
Thank you so much.
All right. Yeah, you're welcome. Thank you guys.
Thanks, Jessica. And before everyone goes, Michael, do you want to give us a brief introduction to our next Chestnut Chat on December 12th?
Yeah, our next Chestnut Chat is going to be really exciting because we're talking to all of our award winners and partner winners and recording conversations with them about their experiences and how it is that they came to Chestnut and what excites them about TACF and their missions and what they've done to earn those awards. So it's a really exciting, and we're looking forward to sharing them with you. All right, everybody. Have a wonderful Thanksgiving too if we don't get to chat before then.